podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. Hey everyone, a great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Keto Citra, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water: heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there. Thank you very much for joining me today.
Thanks for having me.
Do you want to start off by just telling everyone a little bit about who you are and what your lab does?
Yeah, sure. So I'm a biochemist, physiologist with a PhD from Sweden, and then I did a postdoc in Boston in physiology. I started my lab at Stanford where we're studying physiology of peptide hormones and how they can regulate energy balance.
And I want to start, we're going to go do a pretty deep dive into some of this stuff today, but I want to start out basic and talk about some foundational knowledge that I think will set us up for people to understand things that we're going to talk about later on. I want to talk about what peptides are, compare and contrast them to other things, and then get into things like how they're produced and synthesized and how they're secreted, because I want people to understand the spatial and the temporal scale that peptides operate on. And I want them to understand something about how these things are made impacts how we discover them and how much remains to be discovered. So starting out with a basic definition, just tell people, what are peptides and how are they similar to or different than, say, proteins or small molecule drugs, say?
Yeah, so peptides are short chains of amino acids. Typically, think of them as 3 to 50 amino acids in length. So they're short and they act on cell surface receptors, typically GPCRs, to induce cell signaling pathways and transcriptional changes in cells. So this is a broad class of molecules that have widespread functions. They're different than proteins in that they're typically not folded in a structure, and proteins can have many different functions as enzymes, scaffolding proteins, binding partners, growth factors. Peptides are typically seen as signals by themselves.
I see. So they're relatively small. They're smaller than proteins. They're up to perhaps 50 amino acids. So it's a sequence of amino acids, and it acts as a signaling molecule that might activate a receptor, but they're typically not folded into an elaborate three-dimensional shape like a big protein.
That's right.
And so can you talk a little bit about how these things are produced and synthesized, and maybe contrast that with proteins a little bit? one of the things I want to get you talking about is there's quite an elaborate set of mechanisms and processes for sort of pre-processing these peptides before they are fully active and functional in our cells.
Yeah, the process of making a peptide is such an interesting biological process because they're not just made in as small peptides and secreted and that's it. They're typically made, they're encoded in the genome, so they have an amino acid sequence, but they're made as part of the processing of a larger precursor protein. So you have this large precursor protein that is made and then transported into the ER for sorting and secretion. And in there are enzymes, proconvertases, for example, the PCSK class of enzymes that proteolytically degrade this peptide into multiple small fragments that are typically thought of as the bioactive fragments. After the processing, there's also other enzymes that are chemically modifying them. So putting on C-terminal modifications, and then they're stored typically in cord-dense granule vesicles, and then they can be released. So it's quite an elaborate process of making these peptides, and then they're released upon different stimuli.
Mm-hmm. So the sequence of amino acids that the peptide is, it's encoded in the genome. There's a DNA sequence that encodes the amino acid sequence, but it's not merely that the DNA is transformed into the amino acid sequence of the peptide and that's it. Typically, you're making like a large protein and then basically chopping it off in different places and then making some other modifications before the mature peptide is actually fully assembled. So there's a whole sort of processing phase that goes through that's in between the mature peptide and the DNA encoding its sequence.
Right. And it also depends on where you have this pre-processing enzyme, and that is expressed differently in different tissues. So if you take, for example, pre-proglucagon, which is the primary sequence that is making GLP-1 and glucagon, it's actually processed differently in different tissues. So in the pancreas, you get it processed in one way where you get glucagon. And in the gut, you get it processed at the different sites. So you get primarily GLP-1. So from the same sequence, you have this energy conservation where you just start with one sequence, but you generate different peptides in different tissues. There's also this process where you can make a combination of peptides. Oxyntomodulin is an example like that, where you have two peptides that could be cleaved into two different forms, but they could also be combined together. And that has an entirely different receptor and different functions. So you can imagine the diversity of peptides from a single precursor protein.
Right. So basically, you could have one amino acid sequence, one protein, and depending on exactly where it's cut, how it's chopped up, what else is added to it, this can determine the actual peptide that's made. You can get one peptide, you can get effectively a completely different peptide, or you could modify, say, aspects of like where it goes inside of the cell and how it's actually functioning.
Right.
And so there's this whole, let's call it pre-processing apparatus within cells in between the DNA sequence encoding these things and the mature peptides that actually go on to do something. And so I don't think we need to cover off on all of the details here, but maybe people should just appreciate something about how elaborate this process can be. There's essentially something like an Amazon sorting facility that takes care of, packaging things, making sure they've got little barcodes, let's say, and making sure they know where to go and how to function before they're actually sent off to do something.
Yes. Yeah.
And so one of the things that I think we'll come back to too is sort of the diversity and the context specificity of a lot of these peptides. You mentioned, I just want to ask you one more time about this. You mentioned that you can have the same sequence, the same precursor sequence, and it can be processed, the same exact precursor sequence can be processed in two different ways in two different cells, such that the same starting material ends up being effectively a different thing in the pancreas versus, some other tissues, say. What is sort of the significance of that, the ability to sort of process these precursor sequences in a different way in a tissue-specific manner?
It is the significance of that is that different tissue can respond differently to different stimuli. So for this particular example, in the gut, the gut is responding to, for example, a glucose load. So when you eat, you want to have a rapid response where you have GLP-1 secretion from the gut, while the pancreas needs to make a glucagon to counteract this increased glucose levels. So it's sort of a feed-forward mechanism.
Mm-hmm, mm-hmm. And so these peptides are, by definition, they're much smaller than proteins, but they're larger than other things, like, say, a neurotransmitter. Can you talk a little bit about how the size and the structure of peptides relates to the time scales and the spatial scales they operate on? So, in biology, we've got everything from like hormones that might go through the bloodstream and go to all of our tissues and have effects that last for hours and days. And then we've got things like fast neurotransmitters in the brain that are operating, from one neuron to another on the time scale of milliseconds. How do peptides generally fit into this? And how do you start to think about the significance of how long these signals last and how far they travel?
Yeah, so neurotransmitters are typically like seconds, milliseconds or seconds. Larger proteins are typically an hour's time scale, like antibodies that are circulating. And peptides are minutes to hour, maximum 1 hour, I would say, endogenously. So the time scale is shorter because they're degraded more quickly. But the effects on a particular pathway or cell can still be long-standing because they induce different transcriptional responses, for example, FOS in the brain.
Mm-hmm.
So it's not entirely correlated with the actual half-life of the peptide, but you do have something in between a small molecule neurotransmitter and a protein.
Mm-hmm. So they have a half-life that's on the order of minutes. So they stick around longer than something like a fast neurotransmitter, but not as long as something like a hormone. But importantly, their effects can outlast the peptides themselves. So the peptides can be released, they can stick around and do things for minutes, say, but the actual signaling effects they have within cells can outlast the peptide that triggered it.
Yes. Yeah.
And so when we think about the regulation of these things, you sort of mentioned it a little bit there. They're peptides. They're sort of like mini proteins. So there's lots of enzymes that can degrade them. Is that like a purely passive process, or is the degradation of these things by proteases, is that part of the core regulation for how long these signals actually last?
Yes, it's definitely part of the core regulation. Enzymatic processing, for example, DPP4 is an enzyme that is cutting off part of GLP-1, which is contributing to its short half-life. If you mutate that site so that enzyme no longer can do that function, you will extend the half-life. So for sure, that is one of the mechanisms. But these peptides are also very small, so even if you don't have any degradation process in the body, they're still going to be cleared by the kidney.
Okay. So just in virtue of their size, they get cleared out relatively quickly. And so a lot of the focus in peptide research and peptide biology is in the general area of energy homeostasis. So we're going to talk about the GLP-1s. We're going to talk about some other things, but high level to start out here. What is energy homeostasis? What does that term mean to a biologist like you? And what are some of the core concepts there that we want to convey to people before we dig into how GLPs and some other related peptides actually work to affect things like how energy is used in the body?
Yeah, so energy balance is the combination of energy intake and energy output. And that can be maintained in the body through different mechanisms, but that's the core idea, that the energy you're taking in needs to match the output, how much you expend. Otherwise, you're going to either increase your body weight or lower your body weight.
And so these things last for, let's just call it intermediate time scales, as we said, minutes. They can have effects that outlast the peptides that instigated them. I want to talk a little bit about where these things are secreted from and the secretion process itself. I'll let you steer us in terms of the examples that you think are the best to talk about. But when we think about how these things get released at the cellular level, in neuroscience, I often learned that, you have different types of vesicles, different types of secretion mechanisms. Some things get sort of released right away after they're made, and other things get stored and saved for the right time. Where do these peptides generally fall into one class there? Are they sort of secreted on demand? Are they stored up and saved for later? Or does it depend entirely on the tissue and the peptide?
I think to some extent it depends on what the peptide is. So for some of them, you have constitutive secretion. So they're just secreted when they are made. But for the classical ones that we think about in our endocrine organs, pituitary, or the endocrine tissues and cells, they are released upon certain stimuli. So for example, when you have a hyperpolarization or calcium mobilization, then you get this burst of secretion of these stored vesicles with different peptides. But I think the other constitutive secretion is a mechanism that we know less about. And there may be instances where you have regulated secretion through non-core vesicles as well. They're just way less studied.
Yeah. And so when we think about where they're secreted from, you mentioned a couple key areas already. The brain, certain parts of the brain secrete peptides and hormones. As we'll get into, there's gut hormones and gut peptides secreted from the gut. Are there a limited number of tissues in the body that are generally secreting these things, or are they secreted all over the place?
Yeah, so I think for a very long time, the field of endocrinology has really focused on the endocrine tissues. So all the classical ones that you would think of, the brain, the gut, the adrenal gland, the pituitary. But I think we start to appreciate now that these peptides can actually be made from non-classical endocrine organs. Things can be secreted from adipose tissue, from the liver. These are major metabolic organs, but they're not the classical endocrine organs.
Okay, so there's definitely organs that are at least more famous for secreting these things, but over time we've also started to learn that other tissues that we previously didn't maybe appreciate could secrete hormones or peptides actually do.
Right.
And what do you want to say about just sort of the general theme of, I guess you could say one of the purposes of these things is communication across tissues and organs, right? Like if my gut needs to communicate with my brain or vice versa, this is done via some of these molecules that get secreted and travel throughout the body. What do you want to say about that, just to sort of set up and contextualize the conversation that we're going to go into about gut hormones and GLP-1s and that type of thing?
I mean, that's the classical endocrinology view that tissues need to communicate. The brain needs to know what the status is of the cell. How much adiposity do you have? And this is why you have leptin from the fat communicating to the brain. You should eat less because we have enough fat stores, right? The gut is communicating with the brain that we are full. You should stop eating. So it's a very intuitive way of thinking about how these organs are communicating together or with each other. But I think that there's a lot of inter-tissue communication too, like the brain itself communicates a lot within the cell types in the brain. And there's a lot we don't know.
Yeah. And so when we think about neuropeptides and the brain, the hypothalamus is a major center that we talk about here. When we think about how hunger and satiety are regulated, can you say just a little bit about some of the major themes and how the hypothalamus is sensing what the rest of the body is doing, sensing things like leptin from fat cells, as you just mentioned, and then using that to generate signals about what the animal should do with its behavior to meet its demands. And we can keep it fairly high level here, but I want to give people a sense for okay, so the hypothalamus is able to sense hormones and that tells it something about the periphery, how much fat there is, how much hunger should be generated by circuits in the hypothalamus and so forth. But, give us like maybe some examples or a sense of how this works from, the hypothalamus sensing the levels of leptin or something like this to generating behaviors such as, say, feeding behavior coming on or feeding behavior being turned off.
Mhm. Yeah, so I think the most well-known, well-understood process is the melanocortin system, which is a system where leptin acts through. So here you have MC4R neurons. So these are neurons in the brain that release alpha MSH, which is a peptide that suppresses feeding. So you have this loop where you have increased alpha MSH, acts on neurons to suppress feeding. And there are multiple such examples, but the melanocortin pathway is the most well established.
Yeah. So even within the brain, within the hypothalamus, there's sensing of what's going on throughout the body, sensing things like gut hormones, leptin, as you mentioned. But then those signals from the periphery can be transformed into neural signals using transmitters, but also neuropeptides within the hypothalamus to shape the activity that's going to happen in the brain that's actually going to correspond to the animal, going out and finding food or feeling full and satiated and things like that.
Yeah.
And so where do the GLP-1s come in? Let's talk about endogenous GLP-1 first before we talk about some of the drugs that are being more and more widely used now. So endogenous GLP-1, what does that stand for? And can you give us a sense for the major site of production and secretion in the body?
Yeah, so GLP-1 stands for glucagon-like peptide 1. It's one of the bioactive peptides from the pre-proglucagon gene. It's made in the L cells in the gut in response to glucose load or a meal. And it has functions throughout the periphery, but also in the brain. But I would say the consensus in the field is that endogenous GLP-1 primarily has functions in the periphery. So it's an incretin, so it acts on the pancreas to increase insulin secretion to lower blood glucose. So if you have endogenous GLP-1 or just slightly raised levels, you will lower blood glucose, and that's one way of, controlling glucose homeostasis in the body.
Mm-hmm. So the basic idea is the gut is sensing how much glucose is coming in, and the more that's coming in, the more you want to secrete insulin to then, pull that glucose into cells and do stuff with it.
That's right.
And so it doesn't last very long. And so it can get to the brain, but largely endogenous GLP-1 is thought to act mostly outside the brain, in the periphery.
Right. And there are some ideas or data supporting that it can act on the vagus nerve locally and thereby communicating to the brain, but it's thought that endogenous GLP-1 is primarily peripheral, yeah.
Yeah. So there's a couple different ways that something could actually communicate to the central nervous system in principle, right? It can get all the way through the blood-brain barrier and get up into the brain in our heads. But it could also, as you just mentioned, perhaps affect things through the vagus nerve or, nerve terminals that are actually down in the periphery.
Right.
And so endogenous GLP-1, is that something that's also lasting on the minutes time scale? Is it sort of an intermediate signal?
Oh, it has a very short half-life. Within a couple minutes, it's gone.
Got it. So it's secreted in response to basically eating something, and then it's secreted, but it only lasts a couple of minutes.
Right.
And so one of the major innovations of the GLP-1 drugs had to do with that short half-life. Can you talk a little bit about how the half-life of the endogenous peptide is regulated, and then tell us a little bit about how the GLP-1 drugs were engineered to last longer?
Yeah, so endogenous GLP-1 has a recognition site for the DPP4 enzyme that is pretty much inactivating the peptide. So that's one mechanism by which it's, I wouldn't say degraded, but it's inactivated. Mm-hmm. And then it's cleared by the kidneys. And those are the two major modes of, inactivation and clearance. So the way I'm no pharmacology expert, and these are really people at the pharmaceutical companies that develop these drugs. But as far as I understand, You can mutate the site that is recognized by DPP4 and change some other amino acids to prevent further degradation and stabilizing the peptide. And then the major advance was actually putting on a lipid. Conjugated to the actual peptide, which makes it bind albumin in the blood. And that albumin is typically 60 kilodalton. It's large. It prevents renal clearance, which means that this peptide is hanging on to albumin and then secretes it, or like being circulating for much, much longer time, which means that the brain also has high exposure to this peptide for a longer time.
Okay, so endogenous GLP-1, it only lasts for a couple minutes. It's secreted in the gut after a meal. That makes perfect sense, but it's only lasting for a couple minutes. It's going to get cleared out by the kidney. There were some modifications engineered by humans. You add that lipid, you maybe do a couple other things. And the overall effect of that is this thing basically travels and hangs around in the blood longer because it's sticking to albumin. And that allows it to not just last longer, but last long enough to get up into the brain at a higher concentration.
Yes.
And so what do these things actually do in the brain to alter satiety?
Yeah, so different peptides act differently, but if you and there's still, a debate exactly which brain regions are responsible for the function of each of these peptides, because it's hard to determine the primary site of action. As you know, you can have a site where you have activity, and then you have projections to other areas. So for GLP-1, there have been a lot of studies suggesting that it acts on the hindbrain, where you see fast activation in the area postrema, in the NTS, in the hindbrain, and that is then projecting into the hypothalamus, the arcuate nucleus, MC4R neurons, and others. So that is the general idea, and that is how many peptide hormones work actually. Either are directly acting on the hindbrain or directly acting in the hypothalamus.
Got it. So these things get into the brain, and there are neurons in the hindbrain and hypothalamus that have receptors that these things act on. So they're directly interacting with neurons and ultimately changing the pattern of activity of those neurons.
Right. But it doesn't get into the entire brain. I think this is a question that I get a lot. How do these peptides cross the blood-brain barrier? And in many of these cases, they enter through areas within the circumventricular organs, so the hindbrain and the arcuate nucleus in the hypothalamus. So it's not like they're occupying the entire brain. They actually have very specific access to some of these regions.
Yeah. And I think this is an important concept for people to understand. So the blood-brain barrier is, as the name suggests, a barrier between the brain and the blood and the rest of the body. And some things are allowed to pass the blood-brain barrier, and some things are allowed, but they're sort of specially transported across. But then there are other parts of the brain that don't really have a blood-brain barrier. Say a little bit more about that and just really define that for us and where those are at.
Yeah, so there are ventricles in the brain that have direct access to the blood. So the hindbrain is close to the fourth ventricle, and the hypothalamus is close to the third ventricle. And there you can have direct access to peptides that are normally not seen by other parts of the brain, for example, the cortex. So this is a way for the body to communicate directly with those areas in the brain.
I see. So this is basically a feature of this whole system. Is you have the blood-brain barrier to kind of protect the brain from stuff you don't want to get into the brain from getting in. But there are a couple spots where actually the blood-brain barrier isn't really there, near the hypothalamus, near the hindbrain. And it's basically a way for certain parts of the brain to have access to the bloodstream and sample what's in it to help with brain-body coordination and communication. So on the one hand, most stuff in the brain is protected by the blood-brain barrier, and that's a very, tight regulation. But then you've got these sort of special spots that are allowed to sample from the periphery and effectively read what's going on in the body.
Right.
And so when we think about these GLP-1 drugs that more people are taking, there's all different versions of these things. I want to ask you a little bit about some of the new GLP-1s that are out there. So we already talked about one innovation in this realm, which is that the endogenous peptide was basically modified so that it lasts longer and can travel up to the brain and get to the brain at a higher concentration than endogenous GLP-1. Is there anything else that we should understand about how these new drugs based on GLP-1 are operating besides that modification? Is there anything else they're doing in the brain, for example, that endogenous GLP-1 doesn't do?
Yes, we're starting to understand more about, the physiology and endogenous function of these peptides, not just GLP-1, but others too, where we have pharmacology that is good, and that the pharmacology sometimes can be very different than what you would understand by studying physiology, for example, the knockout mouse of GLP-1 receptor. When we look at the basic phenotype, if you're lacking the ability to respond to endogenous GLP-1, you should, in principle, phenocopy a pharmacological action. So, but in the opposite way. But that's actually not entirely the case. So if you remove the ability to respond, you would expect these mice to be obese because they can no longer respond to these anorexigenic signals.
Yeah, you've taken away the ability to read the satiety signal, and therefore they should just eat a lot.
Right.
That would be the expectation.
That is the expectation. And there is some very weak signal that these mice eat a little bit more, but they're not heavily obese, which means that, when you go in with pharmacology, you increase the levels to supraphysiological levels. You're no longer in the endogenous range. And this means that you have effects that you would just never see by studying the endogenous biology. It's true, it's specific because it binds this receptor. And if you remove the receptor, you don't have these effects of the pharmacology, but you cannot see it by understanding or by looking only at the endogenous function. So these pharmacological agents, when they stick around for longer, will now have much different effects than just studying the physiology. And I think that is very interesting. We see already signs that these GLP-1 based drugs can have other functions than just suppressing satiety.
Yeah.
It's increasing satiety.
Yeah.
So they, for example, affect the reward system or addiction, or there are different signals and that you wouldn't see otherwise.
Yeah, and this is maybe an important thing to dwell on for a minute. Simply in virtue of the fact that, the dose is sufficiently high and the half-life is sufficiently long compared to the endogenous peptide, you can get effects that you wouldn't see with the endogenous compound simply because there's now enough time for it to, say, diffuse into places it doesn't normally go to, or stick around long enough that it's activating not just the endogenous receptors like GLP-1, but perhaps additional receptors that have lower affinity for the peptide that maybe the endogenous peptide just never sticks around long enough to act on.
I don't think there's any evidence that it binds to other receptors. I think it's still just one receptor, but you can imagine that there are projections that could happen now when you have a long-lasting signal that wouldn't be there otherwise.
Okay. And so you mentioned effects on reward processing and things like that. Can you say a little bit more about that and what the relevance is here for these new GLP-1 drugs? So how are these, what we think of as hunger and satiety circuits, hooked into reward processing systems in the brain? And what's sort of the relevance to, should we think about hunger and satiety regulation as completely distinct from reward processing generally, or are these things intertwined in a way that we can't unpack fully, that they're sort of really merged together?
It's super interesting, and I think we fully don't know how these systems are related, and if there are pharmacology or even endogenous signals that can dissociate these things. But what we do see is that GLP-1s, at least from our mouse work, and there's also some data in humans suggesting this, that the preference for eating sugar type molecules like sucrose and saccharose goes down when you are taking a GLP-1. So this is not just appetite. This is actually a change in preference. So this is interesting because that ties into, dopamine release, potential reward system. But there are other types of rewards that have been tested, for example, amphetamine and fentanyl, where it doesn't seem to have an effect. So it's really interesting that I think different molecules may do different things. And depending on what neuronal signals and projections that are being sent.
Yeah. So, I mean, that's interesting. It's not just affecting satiety per se, it's affecting food preference. So it's not, just scaling down how much an animal eats. It's actually changing what the animal will choose to eat on top of that.
Yes, that seems to be the overall signal that we see.
And can you give us just sort of a basic sense of what are the major robust effects that these GLP-1 drugs seem to have both in animals and humans? Beyond the fact that food intake goes down, what are some of the other effects that we're seeing on reward processing or reward-related behavior, on hormone release and other things going on that are more endocrine related? What's happening beyond just the simple decrease in food intake?
So it's really hard to say what is primary and what is secondary. You can get a lot of secondary effects, and we know that there's protection in cardiovascular disease. There's lowering of blood sugar. Obviously, that's an on-target effect. There's a lowering of cholesterol. There's all these secondary effects. So I think we need to think about what is primary, made by the signal GLP-1 on the GLP-1 receptor, and then everything else that can be secondary to the weight loss or secondary to the lowering of diabetes, if that is the basal state. So I think in order to answer these questions, all of these experiments need to be done in preclinical models in mice, where you're now doing it in the GLP-1 receptor knockout mice, and also to see what is actually holding up in humans. We have done a lot of mouse work, and although the overall mechanism is highly, highly conserved, some of these behavioral effects may not be conserved between mice and human entirely.
And sort of how the basic effects on food intake and hunger satiety at a high level, how universal are they across individuals? Like, do we see this in more or less all individuals when we look at the animal data? And roughly when people take GLP-1s, roughly what percentage of people do they have the primary intended effect for?
Yeah, so in humans, there's a huge heterogeneity. In our mouse studies and even like non-rodent models, these are inbred animals. So it's very, very low heterogeneity. Every single animal responds about the same at the same dose. So that is extremely homogeneous and robust. In humans, there's a huge range. Some people lose 50 pounds, they lose 25%, 30% of their body weight. Some people are so-called like low responders or non-responders, lose 5% of their body weight. And it's not clear why they're responding so poorly. There has been some studies showing that it correlates with the GLP-1 receptor expression, which makes a lot of sense. If they have less of the receptor, they may respond less, but that may not be the entire answer. And it's unclear why some individuals respond better or worse.
Yeah. I want to ask you a little bit about sort of dose-dependent effects here and how the effects of the GLP-1s can start to change depending on dose. So I would presume, although I don't actually know the answer to this, I would presume there's been a lot of dose-response studies in the animal literature. What happens when you scale up the dose? Do you just get more of the same effect? Do you start to get different effects? Do you get any unwanted side effects when you go sufficiently high? And, I want to have in mind when we have this discussion about the animal work, the fact that, a lot of people are taking these GLP-1s now. And I can only imagine that, if I'm someone who's struggled with weight loss my whole life, I start taking an injection. I'm like, wow, I lost 20 pounds. I should just double the dose. I should just triple the dose. What's going to start to happen if and when people start scaling up the dose? So let's start with the animal data. How much are the effects dose dependent? And do we see them change very much as we escalate the dose?
Yes, so we both have animal data and human data for this. So in animals, it's very dose dependent. So as far as glucose lowering, as far as appetite suppression, it's clearly dose dependent to the degree that the mice will almost entirely stop eating if you're giving high enough dose. And once they come off, they rebound and they start to eat just normally. So it's really an effect of the drug. In humans, we actually know that lower dose will just lower blood glucose, but will not cause weight loss. This is because GLP-1s were first approved for diabetes at a much lower dose. So the Ozempic drug is a diabetes drug. That's a lower dose than Wegovy, which is just the same molecule, but at a higher dose for obesity as the indication. So if you go in with a lower dose, you will have glucose lowering, you will restore your diabetes, but you will not lose a lot of weight. So we clearly know that it's dose dependent and generates different effects. So if you have a low dose, it's primarily effect in the periphery and the pancreas. If you go high enough dose, you will have high enough levels entering the brain, and then you get the appetite suppression. But at these higher doses, you will also have an effect on the gut. We haven't talked much about this, but the GLP-1 also acts on the gut to slow down gastric emptying. It acts to slow transit time in the colon. So this is what is causing GI upset and constipation, and or sometimes both in the same patients. So this is the major drawback of going too high in dose.
Mm-hmm. So these things have central effects in the brain related to hunger and satiety. They also have peripheral effects outside the central nervous system in the gut. So, if you're taking a higher dose GLP-1 drug for weight loss, you're going to be taking enough that it gets up to the brain, affects all of those hindbrain and hypothalamus circuits to make you, let's say, want to eat less and maybe change your food preference and maybe lower the reward value of food and things like this. But it's also physically slowing how quickly the food you do eat goes through your gut.
Yes.
So when we think about that at a fairly high level, let's divorce ourselves from the GLP-1s for a moment. When and why would the body want to modulate how quickly food goes through the gut? What are the advantages and disadvantages of speeding up or slowing down transit time?
So, lowering your gastric emptying time, that makes a lot of sense, because if the stomach is not releasing the food into the intestine, you feel full longer. So that makes a lot of sense. In terms of the colonic transit time, that would just reduce the time that it takes for the food to go through the intestine. I'm not entirely sure why GLP-1s would, what's the evolutionary function of that, but the stomach gastric emptying makes a lot of sense to just increase your fullness. And maybe the colonic transit time is just a functional consequence of that. I'm not entirely sure.
Got it. So when we think about the effects on the gut, the things that we can think about there in particular are how long it takes the stomach to release the food. The longer it sits in the stomach, the more it's presumably going to get chopped up by stomach enzymes, but also just the more full you'll feel. And then there's the time it actually takes to go through all of the intestines and how long it sits in the colon. And so I'm not sure how much is known here, but presumably that would affect things like nutrient absorption, the microbiome, and all of that stuff.
Yes. And I think this is an area where it's still intensely being studied.
Yeah. And so when we think about escalating the dose, so at a relatively low dose, you mentioned, things like Ozempic are basically used as diabetes drugs. That'll be more glucose homeostasis, blood sugar regulation. Basically, the dose isn't high enough for very much of it to get up to the brain to have central effects. But then the higher dose versions of these things are high enough to get up to the brain. That's where you have more direct effects on the central regulation of satiety, and you're going to get more weight loss in general. What happens if you keep escalating the dose further? Is there just sort of a dose regime where you can just escalate somewhat and basically just get more satiety and less food intake before you start to get anything that you would call, say, a side effect?
Yeah, and I think that's very this dose dependent, and it's very personalized too. So different individuals will have different responses. But for sure, when you have central effects, you will experience side effects too. And you see this in individuals that have, in the newspapers, or you look at, they have gotten compounded GLP-1 and then misdosed or dosed 10 times higher. They just end up in the ER vomiting because the nausea is just too terrible.
Yeah. So actually, there was an interview recently with Sam Altman from OpenAI. Did you see that?
Yes.
So for those who didn't see it, Sam Altman, the CEO of OpenAI, he got one of the GLP drugs from somewhere. I don't know where. And he either messed up or the compounding pharmacy messed up, and he took like a mega dose. I don't know how much, but it was like way more than it was supposed to be. And the basic effect was, well, he wasn't hungry and he didn't eat, but he also didn't want to do anything. I think a neuroscientist would call it anhedonia. He was basically just laying in bed, didn't want to work, didn't want to socialize, didn't want to do anything. Is that basically the risk of doing like a mega dose of one of these things, that you're basically just tapping into some of that general purpose reward circuitry and shutting all of it down?
Yeah, I mean that is something that is being studied. The anhedonia is interesting because obviously that's not what you want. You just want appetite suppression, but still have enough energy to do other things. And it's unclear if it's like only linked to the lack of energy intake or if it's something else.
Yeah.
But one thing that we have seen in mice is that we can inject them with high dose semaglutide GLP-1 and then put them on a treadmill. And they can run just fine. There's no issue with like their, energy regulation. They're not exhausted in that they can no longer run. But if you ask them to do voluntary wheel running, so they had the option to run on a running wheel, they choose not to.
Right.
Actually, huge reduction. And why is that? That's sort of an anhedonia response, and we still don't understand what the mechanism is of this, but that's a clear effect that they choose to run less on a treadmill, and I find that very interesting.
Right. So that is interesting. At a high enough dose, they don't choose to run, but they can run. There's no energy deficit preventing them from having enough energy to run. There's no overt motor impairment that's preventing them from like coordinating their muscles. They just choose not to run, and for whatever reason. But that does imply there's some volitional effect there. There's we know that mice do like to run, right? It's a rewarding thing for most mice. They like to run on little wheels and stuff. So that does start to imply that maybe you're tapping into what I'll just call reward circuitry beyond hunger and satiety regulation. And that does start to get me thinking about other things that people are reporting, which is, apparently, and I don't know how meticulously this has been studied yet, but apparently people are reporting spontaneous reductions in things like alcohol intake and drug intake. Do you know anything about that? And does that start to tell us something about what these drugs are doing beyond hunger and satiety per se?
Yeah, so we don't know enough about which areas in the brain are controlled by GLP-1. Those reward areas are typically not thought to be directly exposed to GLP-1, but they might very well be induced or reduced through projections. This is not my area of expertise, but we're working with some collaborators on this, where we're looking at incretins and non-incretin peptides and how they can affect reward pathways and addiction pathways. And this seems to be different responses.
Yeah. So, there could be something really interesting happening here that is also worth thinking about from a clinical perspective, where, you take the dose up high enough on these things and you get weight loss, you get better hunger satiety regulation, and we'll call that an effect that people are after. But then, maybe you go a little bit higher and you start getting less motivation in general. And if you go even higher, then you get, anhedonia and things like that. So, it could be that there's like a sweet spot here where you get enough satiety regulation to give you the weight loss effects, but not so much that it's going to bleed into other domains of life, like just your willingness to do other things.
Right. But I think importantly for the GLP-1s, there isn't a strong signal in, inducing depression. It doesn't seem to be inducing depression, which would be very serious, and that would be a very serious side effect. So it's not just, the dopamine system. There must be something else. We don't understand what that is yet.
Yeah. And there was there was rimonabant years ago. It was sort of like an earlier type of weight loss drug that got released in Europe. And then it was pulled from the market because it was basically causing depression and psychiatric problems for people. And that was causing weight loss and less satiety, but it was acting through the cannabinoid system. So far, it does not seem like these GLP-1s are having that exact type of side effect.
Yeah, that's right. And that the rimonabant story really deterred people from going into this. Like, many people were convinced that you cannot get appetite suppression without affecting all these other systems, and the GLPs have shown that this is not true. So it's really amazing, and has opened up a new area where we can learn more about addiction, reward behavior, and feeding and how they're linked.
Yeah, I want to talk a little bit more about the innovation and evolution of this general class of treatments. We talked already about one innovation, which was, you modify endogenous GLP such that it basically lasts longer and therefore can get to the brain at a higher level. But now there's new drugs that you can talk to us about, and they're not just acting on GLP-1, they're acting on, other things. Basically, we're combining things so that we're playing with multiple knobs in the brain and body at the same time. Can you talk about things like Retta and some of the newest versions of these peptide therapies and what they're doing beyond GLP-1?
Yes. So semaglutide is the GLP-1 molecule by itself. And then there are other approved drugs, tirzepatide, which is a combination of GLP-1, agonism, and GIP. So this is another incretin. They pretty much both come from the gut, and they act in a similar way. It turns out when you combine them, you actually have even stronger appetite suppressing effects. It's unclear whether all these other behaviors that we were talking about are also sustained now when you have the two molecules. And then the third molecule is retatrutide, which you mentioned, which is a triple agonist. So this is GLP-1, GLP, and then glucagon. And this is very interesting because the glucagon component is kind of it's not obvious why this would really work to enhance weight loss even more, because by itself it doesn't induce weight loss, but it increases energy expenditure. That's the equation of you suppress food intake, but you also increase energy expenditure. So overall, you have a greater weight loss and other metabolic benefits. So it will be super interesting to see in the future which of these benefits and side effects will be maintained with all these different combinations. There are some indications that the tirzepatide, for example, has lower side effects, or reduced side effects in terms of the GI upset.
Yeah. So basically, you're talking about drugs that affect one of these peptides, and there's drugs that affect two or even three of them. So we're playing with now multiple peptide-based systems at the same time in parallel.
Yes, but I think it's not clear if these, the triple agonist, for example, has now some neopharmacology that is not seen with the three individual peptides by themselves. That is still being worked out, and it seems to be some indication that it's actually not just the three combined. It's because you can imagine that when you have a triple agonist combined, you will not reach all the brain regions or the same brain regions as you would with the individual peptides by themselves. So you may restrict this triple agonist, or restrict it to certain brain regions, now generating different functional outcomes.
Yeah. So even the triple agonist, it's hitting three different peptide receptors, but there's also additional modifications such that it's also doing things that none of these endogenous peptides do on their own.
Yeah, not through different modifications, but just by the fact that they are fused in a single molecule.
I see. Yeah. So it is literally like a fusion of three peptides.
Yes.
Yeah. Interesting. And so, peptides as a general area is getting a lot more attention lately. And there's all sorts of peptides. One of the things that we talked about up front is, these peptides are relatively small. They're basically mini proteins. And there's also a lot of pre-processing that goes into getting them to their final functional form. And those things sort of imply that there's a huge possibility out there, like a huge diversity of possible peptides. Can you say a little bit about how big do we think the world of endogenous peptides is? And how much of it do you think we've mapped? Have we only discovered a small portion of what's likely out there? And is there a lot more to be discovered?
Yeah, I mean, this is what my lab studies, and this is my entire research program. I think that there are many more that we have not discovered yet. But I would say that, the ones that have been discovered starting from, 1905 and 1909, where the first peptide hormone was discovered, I think we have found the most important ones, like for physiology, but there may be additional ones with functions that we started to uncover now. So I think in it's about a dozen or so that we know fairly well how they work, and they have receptors, and we have mapped out their functions in preclinical models. Some have already gone to humans, but that's less than a handful, actually. So we still have a lot to learn from the ones that we know the identity of. And then my lab is interested in trying to see what else is out there. Since we now have the genome, we can see that just by encoded in the genome based on where these enzymes are cleaving, we know that there should be additional ones that have not been characterized. Whether they're going to be important or not, we don't know, but for sure they're there encoded.
Okay, so you can tell or predict, at least to some extent, just from DNA sequence where a peptide is likely in the genome.
Yes.
And so can you talk about some of the work that's being done there using computational methods to predict and characterize novel peptides? What does that look like? And have you guys discovered any new ones from, just predicting it from the raw sequence?
Yeah, so what we started now a couple of years ago, was to look across the entire human body, how many precursor proteins similar to, pre-proglucagon may we have. So again, we know about a dozen, but we were pretty much using the PCSK cleavage site as a guide, and we're just asking. We know that PCSK1 and 2 are cleaving many of the known bioactive hormones. What else might it cleave that would look similar? And when we did this mapping, we took the sequences in our genome. This is humans, but you could do the same thing in mice. And then just ask which ones have this characteristic signature. And it turns out that it's about 300. Okay, so not all those will be bioactive, of course, but that will tell you where to first look. And then you can divide it up into different organs and see that actually the predominant class of those are in the brain, in the liver, and in the placenta, and then a fewer in the other classical endocrine organs. So this would suggest that there are other precursors that can make peptides that are entirely understudied. And there are also new peptide fragments from known precursors. For example, POMC, there is a fragment that has not been studied. So that would also be interesting to uncover the function of.
Yeah. So I guess at a high level, the world of peptides is much larger than we know right now. There's much more that we probably don't know than we do know, just in terms of the number and identity of all of the peptides that are endogenously present inside of us.
Yes.
And so I want to talk a little bit about some other peptides, but I think before we do that, we should talk about some other things. And maybe we can do this in the context of the GLP-1s and related drugs, some of those that you mentioned. When we think about weight loss and body weight regulation and obesity, right, we've got the energy inside of the equation and the energy outside of the equation. I think the first half is more easy and intuitive for people to understand. Obviously, if you eat less food, that's going to be, part of why someone might lose weight on a drug like this. But you also mentioned that some of these can affect energy expenditure, basically just how much your body is burning through. Can you talk just a little bit about energy expenditure at a high level? and some maybe basic principles there that we should think about. And then I want to get into like, do we actually know how some of these peptide therapies are affecting the energy expenditure side of the equation?
This is a very interesting area, and that's part of, what's missing. So now we have drugs that can suppress appetite. We don't have any approved drugs that can increase energy expenditure. There was a molecule back in the day, DNP, that actually was used in humans. It increases energy expenditure by uncoupling the ATP in the mitochondria, so it creates excessive heat.
Yeah.
So you pretty much sweat and lose your weight this way. But it's also, it causes death, so it's not safe. So that's the whole question: can we increase energy expenditure in a safe way?
Right.
One natural way of doing that is to exercise. Right, so you can increase your energy expenditure by just using much more energy, using glucose in the muscle, and but that's something that it's hard to make people exercise if they're not motivated to do that. But that's pretty much the only way we have currently to increase energy expenditure, or increase your muscle mass long term to have increased basal energy expenditure.
Yeah. Side question. So you mentioned earlier in the animal data for some of the GLP-1 drugs that they would run if they were forced to, but they wouldn't do as much voluntary running. Is there any human data on if these drugs are affecting people's exercise willingness?
Yeah, so I think there are studies, and I think it's complicated to interpret in humans because many in a lot of the human data, the GLP-1 drugs have been given to obese individuals. And as they're losing weight, they start to move more and they're more motivated to exercise, or they have increased movement just in general because it's easier to move around. So I don't recall any strong signal in humans that it's causing less voluntary exercise.
Yeah, so I imagine that's actually pretty
unclear if that's like just a mouse phenomena or if this is really true in humans.
Yeah, I imagine it could be pretty difficult to parse that too, because it's like, well, is it a direct effect of the treatment, or are they getting more positive feedback in their life because they're starting to lose weight and they just feel more motivated? It might be difficult to tease that out.
Yes.
Yeah. And so there's lots of other peptides that people are taking outside of the context of being prescribed them by their doctors. And there's all kinds of peptides out there that people are talking about in different ways. Some of them that I've learned about on the podcast and elsewhere affect things like energy expenditure. But I do want to get sort of your perspective as a peptide researcher on the social phenomenon of peptides right now. In thinking about the success and the wider spread use of GLP-1s, thinking about how many peptides are out there and the more attention that's on them right now, and just thinking about like the full landscape of how people are allowed to use these things, not allowed to use these things. They can order them online without a prescription, but they're not supposed to be injecting them, but they are. What is your sort of general take on this phenomenon as a peptide biologist? Is this relatively exciting? Is this kind of scary and dangerous? Are these things, I guess one question would just be like, how dangerous do you think it is to be playing around with peptides as an individual?
Yeah, overall, it's quite scary because peptides can be very, very potent, both, the on-target effects, the real effects of them, but also the way that they're made and administered, it creates a lot of risk. But in general, I think the impression is that because peptides are endogenous molecules, they're made by the body, they are generally safe. And I think although that may be true that their physiological function is safe, we're talking about pharmacology where you're injecting a very high dose. So some of these newer peptides, for example, melanocortin, is injected to suppress appetite, increase tanning, and have all sorts of other effects. It's actually acting on all the MC receptors, so MC4R, MC1R, and MC3R. It's a polyagonist. So it's it is an endogenous similar molecule, synthetic peptide made from an endogenous molecule. It acts truly on those receptors. It's specific, but because it's such a high dose, acts on multiple different receptors, you will have a lot of side effects. And I think sometimes the general public, they don't really appreciate that.
So melanotan, named after the fact that it's interacting with all of these melanocortin receptors. We talked about melanocortin 4 earlier, but there are a variety of these melanocortin receptors. They're very important in the body. And its primary effect that people are after is that it makes them tan?
Yeah.
Yeah. And do we know anything else about side effects, known side effects or expected side effects from playing with all of those receptors?
Yeah, so it changes, the melanocyte production. So you can develop like nevi or hyperpigmentation. It also suppresses appetite and can cause nausea. So a lot of side effects associated with the true effect of these peptides. There was some speculation that it can increase your risk for skin cancer. I'm not entirely sure if that is true, but again, it has not been tested. Like, we don't know what the long-term effects are. So even if you manage to get an entirely pure peptide, it's not a contaminant in there, and it's purified in the correct way. Even that, it needs to go through rigorous testing. Like, I wouldn't take it.
Yeah. So there's the question of, are you actually getting the pure peptide? And assuming you are, then what is it doing? And then there's just the simple fact that there's basically no long-term human data on any of these, more or less.
Right.
What are some of the other popular ones out there that people are talking about and taking more, such as melanotan, that you've heard about or know anything about?
There's this BPC-157 molecule, which people take for like muscle recovery. And that has come up in multiple different scenarios, and people have asked me about it. So I did a quick search in the genome, and it actually does not have a precursor protein. It's described as a gastric peptide, identified in humans. It actually does not have a human sequence. I don't think it exists. I could not find any homolog or anything similar to this. I think it's just probably a contaminant that it has been reported in the literature, but it was I think it's probably just a contaminant. You can make this peptide, you can inject it. Maybe people see something or not. It does not seem to be reproducible. People still take it for various things. If it has some inflammatory effect or some off-target effect, I'm sure people can see something or observe what they think is a true effect, but that is pretty much entirely unfounded as far as the scientific evidence.
I see. So BPC 157, body protection compound 157. If I was hearing you correctly, this was originally isolated from like a gastric extraction of some kind. And so it was speculated that perhaps it's an endogenous peptide or derived from one. But you're saying that you did a genome search using some of the tools you've developed, and there's no obvious indication that there's an endogenous version or precursor to this in the human.
There's no endogenous version. Yeah.
Okay. And that would be would that be pretty clear for you to see if it was there?
It's absolutely clear. Yes.
Yeah. Okay. So it's a peptide. You can make it. People are using it, but it's not an endogenous peptide.
Right.
Got it. What about, so one of the other ones I've heard a lot about, and I've done a little reading on in terms of the animal literature, which is very interesting actually, was MOTS-C, this mitochondrial derived peptide, which does seem to have, in rodents at least, an anti-obesity effect on food intake, and also does seem to affect energy expenditure. Is this something that you know much about?
I don't know much about that, the mitochondrial peptides, but I know that has been a big area of investigation. This is not the only one that has been identified. There are other ones too that are these cryptic peptides or peptides that are mitochondrial that have been missed sort of in the genomic mapping because they were typically shorter than what we would assign a gene.
What are some of the major projects that you're working on in the lab right now with regard to peptide biology that you're particularly excited about?
We are working primarily on trying to identify non-incretin peptides with biological activities. So things that don't act on the pancreas to increase insulin secretion, but rather act through some other receptor or mechanism to control feeding or to control other aspects of metabolic regulation. But primarily, we're looking at feeding behavior.
Mm-hmm. And you did, there is a paper that I glanced at where you discovered this thing called BRP. So can you talk a little bit about how you discovered that? And then maybe a little bit about what's the motivation to find things like anti-obesity peptides that act through different mechanisms from the GLPs?
Yeah, so the reason we were looking for non-incretin and non-GLP type peptides was we wanted to have a way where we modulate feeding without affecting the gut. So where you don't have any effect on gastric emptying, or that would be one way where you can imagine that you would avoid the side effects on inducing nausea and GI upset. So that was our motivation for starting that. And we did first a computational search across the entire human genome to look for pro-hormones. And then we focused on the brain. So this BRP peptide is the precursor, is expressed in the brain, in the cortex, but also in the hypothalamus, and has pretty high expression. And it's expressed in cells that also express the PCSK1 enzyme. So it's possible that since it has these canonical cleavage sites, that it's making small peptides. So we saw this, and what we decided to do was to generate small peptides and just test from this parent protein. It's called BRI-NP2. And then we tested them by injecting them into mice and look at different physiological functions, including feeding, glucose regulation, and other things. And what we quickly saw was we tested, I think, 20 or so high potential candidates in mice. We injected them, and this BRP peptide really robustly reduced food intake. Within 30 minutes, the mice stopped eating.
Okay, so this was an example where you basically predicted the existence of this from the DNA sequence, and then you went and looked, and indeed there's this BRP peptide, and it has an anti-obesity effect basically, but it's acting through a different mechanism than GLPs.
Yes. So when we have done now, we did experiments, for example, in the GLP-1 receptor knockout mice that no longer respond to GLP-1. They still respond to BRP. So we know that it's an independent pathway. And we can also look in the gut, and we don't see any gastric emptying effect. So there's no slowing down of gastric emptying. So it only acts on the brain. And when we go into the brain, we can do whole brain phos mapping, and we see that it activates different neurons that are non-overlapping with the neurons that are activated by GLP-1.
Oh wow!
So we can clearly see it is an activity. There is an activity, but it's just not in the same neurons.
Yeah. So the GLP-1s have central effects in the brain and peripheral effects in the gut, one of which is they're slowing transit time of food, basically. This new BRP peptide that you guys discovered using computational methods has an anti-obesity effect, a weight loss effect in rodents. It seems to have no peripheral effect, or at least not the gastric emptying and transit time effects that GLP-1s have. And it does seem to act centrally in the brain, but it's doing something very different in the brain than GLP-1s. It's affecting different neurons in different ways.
Yes.
Mm-hmm. So I guess the hope here would be that you could discover peptides, develop new peptide therapies that either A, work for people that don't really respond to GLP-1s for whatever reason, or B, do respond to GLP-1s, but maybe have side effects that they don't like.
Yes. And one idea, I mean, we're not trying to compete with GLP-1s. GLP-1s are absolutely amazing. That would be first-line therapy for a very long time. And I think it's an amazing discovery, an amazing drug. It doesn't work for everybody, and you can imagine that some people need to go off of it and need to have other types of therapies, or you can imagine that some individuals would need even more weight loss, where you can add on different other types of peptides.
Mm-hmm. And so endogenous peptides, as we said, operate on intermediate time scales. So not seconds, not hours. They're more operating on like the scale of minutes. So they don't stick around forever, but they last longer than some other super fast signals like neurotransmitters and things like that. the engineered weight loss drugs like the GLPs are engineered to basically last longer. Is that is sort of the timescale that endogenous peptides work on? To what extent is that a feature or a bug that we want to engineer away? Do you think a lot of these peptides might be best used as like short acting things? Or do you think like we're going to continue to need to modify them to make them last longer? And do you think that could pose any downside risk in terms of, what these things are going to end up doing to people who use them for years and years?
Yeah, so I think it really depends on what molecule it is and what the function is of that molecule. So we have had insulin for over 100 years, and this is a molecule that you want to have very short acting, and people inject insulin. It does its thing after a meal, lower blood glucose. And they're even looking to make even shorter acting insulins. So it depends on what kind of effect you're looking for. It may not be beneficial to extend the half-life of, all the molecules and all the peptides into a long-acting molecule. You may induce other types of effects or other side effects you wouldn't see if it would be short-acting. But I think it's more a practical question. So before semaglutide and Ozempic came along, there was a molecule called liraglutide, and they had to be injected daily.
Yeah.
And that's just a thing that people don't like to inject daily if you can inject weekly. So all these new drugs that are coming along, there are like antibody conjugates, different things that are put on the peptide molecules to make it, a monthly injection, or that typically sounds more attractive. But not even talking about the oral peptides that are coming along now.
Yeah. Well, that's great that you brought that up. That's something I wanted to ask you about. So you mentioned BPC earlier, which was derived from a gastric secretion, which implies that it has some ability to withstand like the stomach, basically. Given the fundamental structure of peptides here, they're just small sequences of amino acids. How robust are these things, or can they be made to withstand the GI tract so that oral routes of administration are viable? Are most of them going to be chopped up very easily in the stomach and inactivated? Or is it relatively easy to discover or engineer peptides that are going to be orally active, and then there's no need for injection?
For a very long time, I think the main hypothesis was that we cannot make peptides orally bioavailable. But it has now been proven that we can actually do this by engineering the actual peptide, or by administered with things that protect them from degradation. But in general, it's not an easy thing to do. It really depends on the molecule. You may not be able to do it at all with all the molecules that has been shown to work for GLP-1. It may not be applicable for all these other duogonists and triagonists. We'll just have to wait and see.
And how long now do we have data for on long-term use of GLP-1s in humans? What's sort of the longest time frame people have been studied in a clinical setting who have taken these? Because one of the things that people talk about is you often need to stay on these things to maintain the weight loss. Some people are probably just going to stay on them for years and years at a time. That's likely for a large number of people. Is there any reason to believe that you might have negative consequences that only arise after very extended periods of use? And how long is the clinical data that we actually have in terms of the length of use right now?
Yeah, the clinical data for GLP-1s at the dose where they're treating obesity is not very long, but they have been used in individuals with diabetes for a very long time. 10, 15 years now. And it only seems to be positive so far. So there hasn't been any direct long-term negative side effects. If anything, seems to be protective against cardiovascular disease. And there are some indications that it has a protective role against certain types of obesity-associated cancers even, although that data is still coming in. We will have to see what at this higher dose, whether it will be even larger benefit or whether there will be some other side effects. But in general, they're thought of as beneficial.
And so what are some of the other projects that your lab is pursuing today beyond the BRP story?
So we're working on other modifications of peptides. So we have done a similar search where we look for potentially new cleavage fragments with imprinted, modifications. Phosphorylation is a modification that is typically thought of as being put on proteins, and that is very well established that it affects the protein function. On peptides, it also is there, it's detected, it has pretty much no functional value as far as we know. We think that there are some scattered papers showing that phosphorylation of a peptide can affect the bioactivity, but it's not entirely clear how that works. And we find that there are many hundreds of peptides that are phosphorylated on a serine. And how does that affect the function? It's entirely unclear. So we're working a lot on trying to understand what is the rest of the landscape of these peptides. When are they made? Where are they made? How are they modified in a sort of larger scale than what was done previously? In the 50s and 60s, they went one by one, purifying them and looking at what particular modifications were there. We're trying to do this in a more systematic way and then try to identify which ones of these are important.
So a lot of people are using a lot of different peptides at this point, as we've touched on. There's also going to be some potential new regulatory changes coming up this summer that affect basically how easy it's going to be for people to acquire these research peptides. As the type of scientist you are with your background in peptides, what is your sort of general make of that in terms of what you're going to be looking out for and what you think people should look out for when they think about accessibility and actual personal use of these things? And I would love if you could do that sort of in the context of just like the stability and the structure of these things and how easy it is to store them, to make sure they are what they say they are, and just how stable and safe they are as molecules that might be sitting in someone's refrigerator.
Yeah, I would be very careful just in general to take anything where you don't have an analytical instrument to really make sure that what is in the vial is really what you think it is. I think for many of these research grade peptides, we buy them for research use. And the purity can be anything from like 85% up to 98%. But what is the rest of the, even if it's 98% pure, the rest of it could be something else that you have no idea what effect that could have. Injected at a high enough dose, that could actually be significant. The other thing with research grade peptides is that they're often dissolved in different types of salts. So there's a TFA salt that is very often used when you make in just general peptide synthesis. That is not safe for human injection, and it's pretty much impossible for a general person they don't label on the vial whether it's made with TFA salt or something else. So it's pretty much impossible to know what's in the vial. And then again, if you dissolve it in sterile water, how do you know that it's actually sterile? How do you know that you store it in the fridge that is not aggregated? That is a major concern that was a problem for amylin peptides for a long time, where the endogenous peptide is actually forming amyloids and form like aggregates. And if you inject this aggregate, it can accumulate in your tissues, in the pancreas, maybe in the brain, and cause some severe adverse effects that you don't know what the functional consequences are. That we have no idea about for any of these new peptides. So this is short term. It may not cause any harm. You say, "Oh, this is safe. It's fine," and I injected and nothing happened. But you don't know long term what the chemical structure of this peptide looks like. You would need a very advanced instrument like an NMR to determine if this is aggregated or not. If it's not sterile, that's a whole other issue, right? You keep it in the fridge for some time, you take it in and out. Maybe you're not using sterile technique to draw up in the needles, and then you're injecting yourself with this. You can cause sepsis, right? So it's just not safe.
Yeah. So if I'm hearing you correctly, even when you guys in the lab order research grade peptides for literal research purposes, they can be of variable purity, and it's very common for them to be 98% pure or less, and you don't even know what's in there, the other 2%.
Right. I mean, we do analysis. We do analysis of them to see what's in there. So we use LCMS or NMR to look and see what's in there. But unless you know how to interpret that data or have access to this instrument, how would you know?
Right. Yeah. So if someone buys peptides online and it's 98% pure, maybe even the company that made it did test it, but they probably don't even know what the other 2% is, and that's just going to be a question mark.
Right.
Yeah. One of the final topics I want to ask you about, and this is something that people are also talking about with respect to the GLP-1s, is what's going on with the weight loss in terms of the tissue that's lost. Is it mostly fat? Is there lean mass being lost as well? How does that compare from lean mass loss and fat loss that you would get from regular dieting? And things like that. So can you just kind of give us a lay of the land on what the data says with the GLP-1s and related drugs in terms of fat loss and preservation of lean mass?
Yes. So there's been a lot of discussion in the field because there have been studies in both humans and mice showing that when you're losing weight, when you're on a GLP-1, you lose both fat mass and lean mass. And there's a concern that the lean mass loss would lead to reduced muscle function or sarcopenia. But I think the latest data would suggest that you don't lose predominantly more lean mass when you're on a GLP-1 versus if you're just dieting and go on caloric restriction, you lose body weight, you lose the same proportional amount of fat mass and lean mass. A leaner person will have less lean mass. That's not necessarily detrimental. So unless you have good markers or good ways of measuring actual muscle function, it's not clear whether this is actually a problem or not.
Got it. So if you're going to lose a substantial amount of weight, you're probably going to lose some combination of fat and lean mass, such as muscle mass, regardless of how you do it. So it's really a matter of, are these drugs causing more lean mass loss than you would get from regular, dieting weight loss, above and beyond what you would get from that? Or is it about what you would expect from doing that? And you're saying basically so far, it doesn't seem to be an obviously concerning amount of lean mass loss.
Right. And when you look, so for the preclinical studies in animals, if we look at muscle strength after GLP-1 treatment, the muscle strength is actually higher per milligram of muscle tissue you have. So even though you lose muscle mass, you actually don't lose muscle strength. So the strength is intact, which would suggest that the muscle is working just fine. But this is in, young animals. It may be different if they're very aged. So and I think that this is true also in humans, that there's no reduction in like grip strength, and there's not really good measurements in humans, but it doesn't seem like overall muscle function is reduced. But this is the average, I think, GLP-1 user is like a 45-year-old female. So it may be different in males. It may be different in very aged individuals that are losing a lot of weight, that are not exercising. So we would need to really look at different populations and different amounts of weight loss, and maybe their protein intake. So that is something people talk about when you're on GLP-1. You should also take extra protein to see if there's any clinical detrimental effect. So far, it doesn't seem to be that the evidence is suggesting that, but it could be a potential problem in, elderly sarcopenic individuals.
Yeah. So I want to ask you a question from your perspective as a scientific expert on this stuff, but not as a physician. So we're not giving medical advice or anything, and we never do on this podcast. But let's imagine ourselves in the shoes of someone who's kind of like that average user that you mentioned. You're middle-aged, maybe you're in your 40s or 50s. You're overweight, but you're not morbidly obese, you're not catastrophically metabolically ill. You're just hoping to lose some weight and get into somewhat better shape. You've struggled somewhat with that, but you've probably put in, say, a normal amount of effort. You're sort of a normal potential candidate for one of these things. What would be the kind of calculus in your mind that you would go through to evaluate whether or not to try one of the GLP-1s that's out there if you were such a person?
I think this it's a personal preference, but also these, unless you're talking about research grade drugs, which is not recommended for anybody, if a physician is prescribing these compounds, then I think following the advice of them would be the best way to go. I think I don't have any strong opinions either way. Whether they should be used, whether you're clinically obese or not.
Okay.
I think the BMI scale is different for different ethnicities and different populations. So you can also imagine that there are many scenarios where you're not morbidly obese, but your values, your LDL, cholesterol, glucose levels, everything would be improved if you would just lose 10 pounds of body weight.
Yeah. So you can imagine situations where it would make sense to take it from a metabolic health perspective, even if you weren't morbidly obese. And then how would you, let's just talk a little bit more, one more time about the side effects. What are the major side effects? And are those generally things that are going to be, you think, relatively mild for people, or things they should really stop and think about?
The side effects will depend on the dose, and actually it will depend on the individual. Some individuals report very few side effects, and some have to stop taking them because they cannot stand the side effects. So it's hard to predict how you're going to respond.
Mm-hmm, mm-hmm. Is there any risk that, in general, physicians might get a little bit too enthusiastic about prescribing these things, as they perhaps had for other drugs in the past?
It's really hard for me to speculate on that. In general, I think they would prescribe according to the guidelines.
Okay, that's an interesting answer. Any other exciting projects that you're working on in the lab that you want to talk about before we stop today?
Yeah, so we're working on weight loss drugs and their potential benefit for cancer. And I'm really interested in this area just because of the clinical data that we see with GLP-1s, and that may be protective against certain types of obesity-associated cancers. And we're extending this now to look at other molecules that are reducing body weight. Is this directly correlated with the body weight loss, or is this something else in general? Anything that is causing weight loss, would that be protective? Because that's another layer that where we could have a huge benefit for a large number of people. The other area where we're also entering into, although it's not my area of expertise, is the addiction and the reward pathways. Mm-hmm. I think many of these molecules are made in brain, and they are made in different areas, and they gotta have some function there. And can we identify some common circuits, or can we identify something where we can modulate them with pharmacology that is mimicking the endogenous function? I think that would be extremely interesting.
Yeah, so kind of similar to like the idea of can we develop therapies that speak to the hunger and satiety regulation without hitting reward processing more generally? This would be, can we develop something that might be good for addiction without bleeding into some of these other things?
Yes.
Yeah. Interesting. Well, I want to thank you very much for your time. We covered a lot of ground. Is there anything else you want to say? Any final thoughts or anything you want to reiterate about peptides or GLP-1s generally before we sign off?
No, this was a very interesting discussion. Thank you.
All right. Well, Dr. Katrin Svensson, thank you very much for your time.
Thank you.
Podcast podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content.